The Adventurous Life of a Carbon Atom by Alicia Juliet Axelius. Ouch! That was so hot! Why did he have to burn us all? Everyone from our underground squad is here now, in carbon dioxide. There's so many more, and all of them are talking about the numbers. That they were too many here already, and now me and my underground buddies are here too. This isn't good. We were all forced out from under the protecting ground with these big, horrendous machines. They burned us up, and now we're out in this crowded atmosphere. I like it down there. It's way better than out in this dangerous world. By the way, I haven't been here in a million years, literally. It looks so different. What are those creatures walking on two legs? There are so many, and they have really changed in nature. It was more green before. Now it's all gray. Whoa! In an instant, I was sucked into the leaf. Then I continued to one of the cells and into one of the organelles, the chloroplast. Me and 6 other molecules were sucked in, one at a time, into the chloroplast. I really do not like being in here. As you know, the chloroplast is filled with a thick fluid, the stroma. It is horrible, thick and slimy and incredibly hard to move in. Not at all like the air. We can move and flow freely. This reaction is short though. I will survive. Oh, here comes my ride. Hello, who are you? Hi and welcome. Right now we are RUBP, the very first molecule of this reaction. But as soon as you join us, we become a very, very unstable 6 carbon molecule instead. So hurry up and hop on. Ah, this is so unstable. It will break any second now. And it did. What was that? Those were just ATP, and these are NADPH. They are energy boosters, giving us energy. They were activated by sunlight in the light-dependent reactions, and then transported here to the Calvin cycle, which is the name of this part of photosynthesis. Also, if you didn't know, we are now PGA, or 3-phosphoglyceric acid. Oh yeah, I remember from when. Enzymes! They're making me all dizzy. And we became PGAL. Where are we going now? I'm so happy because I think we were just chosen to become glucose. Woohoo! What? Bye, moles, and good luck next time. Goodbye. What did you say about us being lucky? Are we glucose now? Yes, we are glucose. And do you know what that means? Well, of course you don't. It means that we will soon be released from here. See, glucose is one of the products of photosynthesis, which means that it is released. Only 2 out of 12 PGLs are chosen to become glucose. And those two are us. So we will be released. But what happens to them? Those 10 will soon be transformed back into RUBP and repeat the entire cycle one more time. That's what has happened to me, and it feels like I've been here forever. You are extremely lucky. You only had to repeat this one single time. Okay, now I understand why you're so happy. Well, what are we waiting for? Let's go. Now we're finally on our way to freedom. What? We're stuck. And we're not glucose anymore. Did you know anything about this? Bad news. We're not getting out yet. How can I be so stupid to forget? After glucose is made, it is usually converted into starch and then stored. Oh no. For how long are they stored? It depends. Hopefully we were not placed in the tubers of the plants, but in the fruit or the seeds. Then it won't be long. We were in fact lucky. And the red apple was eaten. Oh, now I'm inside of one of those creatures again. That was so much fun. Yeah, I want to go again. I don't. It made me so dizzy. Is that what I think it is? I'm afraid so. No, not a cell again. That was another one of those ATP moles. And now we're PGL again, right? Yes, that is totally right. We're now PGL and Anne is second. We are transformed into biphosphoglycerate or BPGA with a little bit of help from two NAD plus molecules, which are similar to ATP. This is so exciting. I never have any idea of what will happen next. We are transformed from one molecule to another. It is so much fun. Yes, it is. The first time. By the way, the reaction we are in now is called cellular respiration. And it is a way for living organisms to take advantage of the chemical energy stored in glucose. They do this by converting it into about 36 ATPs. It involves three main reactions. First is glucose lysis, which we're in now. Then the Krebs cycle, and finally the electron transport chain, which I know nothing about since carbons are not allowed in there. Glycolysis is anaerobic, meaning that it does not require oxygen. It involves a bunch of transformations between molecules, as you have seen, and though it uses 2 ATPs in the beginning, it has a profit of 2. However, we do have 34 more to go. I think I remember. So what's next? But I couldn't say much more because things started to happen. First came two ADPs and converted us into phosphoglycerate, PGA. And then came two more and made us into pyruvate. Pyruvate is the last molecule of glycolysis. So me and my friend were moved into the mitochondria to get ready for Krebs cycle. So, I guess this is the Krebs cycle. Actually, we are not in the Krebs cycle yet. First, we need to undergo the so-called prep step. As you might understand, this stage is the preparation for the cycle. See, we are not able to enter as pyruvate. We will become something much bigger. We will? Like what? It is a very big molecule with a pretty long name. It is called acetyl coenzyme A, or acetyl CoA. Oh, how big is it? You'll see. And at the same second, a big molecule entered and bound to us. Hello there, I'm currently pyruvate. Who are you? Oh, hi, I'm coenzyme A, CoA for short. Nice to meet you. Nice to meet you too. I heard we will soon become acetyl CoA. Sounds almost like your name. I know, that's why I'm here. With some help from NAD plus molecules, we will be converted into acetyl CoA, and the NAD plus will be reduced to NADH. However, during the reaction, one carbon and two oxygen has to leave us as carbon dioxide. Oh, I hope that is not me. Though I want to be free, I'm still curious of what a Krebs cycle will be like. Yes, I'm finally free! Feels like I've been here forever. Oh, I can't wait to get out. It would have been fun to repeat this one more time though. Goodbye, Moles. We will miss you too. And we were finally sent to the Krebs cycle. Are you ready for the Krebs cycle? Maybe? Yes, so you know, there will be a great deal of transformations between many intermediates. It is very complicated and confusing. Oh, should I be scared? No, you'll be fine. It all starts with a reaction with a 4-carbon molecule, oxaloacetate. The big coenzyme is released and we become citric acid. Since it is the first molecule of the cycle, some moles call it the citric acid cycle instead. I think I still like Krebs cycle more. I agree. Oh, get ready. Here comes oxaloacetate. Okay, so bind, react, release. Well, that wasn't so bad. I liked it. I did too. This part isn't so bad, but it is just a beginning. And a minute now, the irritating intermediates, as I like to call it, will come. My friend was right. It sure was so confusing. There were a lot going on. NAD plus is coming in, reactions occurring, and as results, carbon dioxide molecules kicked out. And one of the carbon dioxides was me. Again, I was released out from the stromata. Again, I was carried through the body of the creature. Again, I was free.